Okay. Hey guys, thank you for joining us. This is our open house event where we're going to highlight equipment at Georgetown University. So we try to do maybe 2-3 open houses per year, and basically the purpose of those events is to highlight our services, and then basically also help you to join I guess those services and provide you with support if you need the support. So I think with this I'm going to switch to, I guess, okay, Marta, and then yeah, thank you.
Thank you. Good morning, everyone, and thank you very much for being here. Today we are highlighting a new technology available at Georgetown University. We have Dr. Andrew Quong, who is the Scientific Chief Officer of Fluidigm, who is going to present this new technology in which you can combine imaging mass spectrometry. And I'm gonna, Andrew, thank you very much for being here. And the stage is all yours.
Great, thank you. Let me go ahead and start to share my screen. Let's see if make sure I can do this. So I hope you can see my screen. Let me go into presentation mode.
Yes, perfect.
Okay, I hope you can see this. So what I will do is I'll spend about 30 minutes, and if you have questions along the way, please feel free to stop me and ask questions. What I'm going to talk today about is the mass cytometry platform that Fluidigm offers that can be used both for doing imaging mass cytometry, which we will be talking about, as well as suspension-based analysis of single cells. And so I'm going to start by giving kind of a brief introduction to Fluidigm. Fluidigm has been around for about 20 years. You may have known us more for the things that we've done in microfluidics. We really have pioneered the microfluidics technology for doing things originally in the area of X-ray crystallography and crystallization of proteins. And more recently, we really have focused that technology on the use of genomics applications. So that would be next generation sequencing library prep and PCR. About 5 years ago, we acquired a company called DVS that was based out of the Toronto area in Canada that really allowed us to bring some additional single cell technologies to the market, particularly in the area of suspension-based mass cytometry. So really the goal of Fluidigm is to develop life science tools that can be used to improve life through comprehensive development of health insights, and more recently, also the development of new diagnostic tools for disease. This is just kind of a quick snapshot. It's actually probably about 6 months out of date. Imaging mass cytometry is a relatively new technology. It was I think the natural evolution, and we'll talk a little bit about that, of our suspension-based platform. And we launched our first commercial instrument in about 2017. And since that time, we've seen significant uptake and a lot of adoption of this, primarily across academic centers such as Georgetown. We actually are at well over half of the NCI designated cancer centers, as well as in 9 of the top 10 pharmaceutical companies. And these are just a couple of examples of some papers that were presented relatively recently at a symposium that Fluidigm had sponsored. So to really think about what mass cytometry is, if we kind of start with the box that's above 3, that's kind of hidden in the back. That is our Helios mass cytometer. And you can think of mass cytometry very, very much like you think of traditional flow cytometry, in that you are labeling single cells with antibodies, and then you analyze each cell individually. And in the case of flow cytometry, you're looking at the readout being photons. So you're looking at the expression of the different proteins by the amount of light emitted by the particular dye that you're using. Now, of course, you're all familiar with the issues around fluorescence. One is, of course, you can...
I'm sorry, Andrew. It seems that your complete slide is not appearing. It's a little bit cut.
I don't know why. I can't really tell.
So it's people is, can you start again the sharing? Just
Sure,
maybe.
let's try that. Let's see if that's better. Is that any better?
Yeah, perfect.
I don't know why it did that.
Yeah. Yeah.
Wonderful world of Zoom these days. So instead of labeling the, measuring the optical output, where with all the different dyes you can have spectral overlap between them, autofluorescence, and simply problems around quenching. The approach that mass cytometry takes is that you label your antibodies with different metal isotopes, and then rather than essentially counting photons, what you do is you count the metal ions using a mass spectrometer. And so that's the advantage that you have with mass cytometry, is that you don't have things like spectral overlap. And so doing things like panel design, where you don't have to optimize the choice of the dye and the antibody, for that specific cell population that you're trying to measure, makes design much simpler. In addition to that, we can very easily and very routinely measure 40 or 50 markers in a single experiment. And I can show you some examples of how some of our customers have used this technology in looking at very specific answers, or trying to answer very specific questions in different areas of immunology. And so we also offer a large number of conjugated antibodies, both for imaging and for suspension. And this makes things very, very easy to get going. And I'm going to show you, I think, an example on this next slide of some of the examples of people using our products around immune profiling, especially in COVID-19. I'm not going to go through all of the examples here, but one really key feature of the use of mass cytometry compared to flow cytometry is with the products that we have developed, you can actually do a very large clinical study across multiple sites with different operators, with different lots of reagents, and get incredibly low CVs on your cell populations. And I'll show you some data for that in a minute. And in fact, this technology has been adopted by a lot of different groups for their standard for doing immune profiling. And so the PICE group has adopted this technology and developed a standardized 45 marker panel approximately for their studies. And then there was a very large study that was sponsored by the National Institutes of Health, looking at the long-term effects of SARS-CoV-2 infection and those individuals that have severe COVID-19. And this is a one-year longitudinal study that goes across 10 clinical sites. And because of the low variability in the results that you get from from mass cytometry, they chose this particular solution. The MaxPAR direct immune profiling system was the system that was chosen. And what's unique about this product is that there are 30 markers in the panel. And we have the ability, which I'll show in a subsequent slide, to run additional to add additional markers on top of that. We also have a proprietary data analytics tool, that allows one to normalize data and essentially do data analysis from this 30 marker sample in about 5 minutes. And these 30 markers uniquely identify 37 immune cell populations, which shown on the wheel are the different markers that are used in this panel and the different isotopes that we use in this particular assay. So the way the assay works is the antibodies are actually freeze-dried and are sitting in the tube, ready to go for your labeling. And so we actually offer 2 different processes for with this kit for doing the staining of the cells. In the first, it can be just 300 microliters of whole blood. So you'll add the blood to the tube, incubate, and then basically lyse the red blood cells, and then do a few washes. And then you can essentially, in about an hour or 2 hours, have your sample ready for analysis on the mass cytometer. The other way, of course, you can do this is you can do this with frozen or fresh PBMCs. And in that case, after you've isolated your PBMCs with your choice of method, again, the staining takes about a couple of hours to do, where you basically just add the PBMCs, add the appropriate buffers, incubate, and then wash, and then basically load your stained pellet onto the mass cytometer. And so what I'm showing here on a larger version of this slide are kind of some of the different T and B cell subsets that you can identify with this particular panel. And as I said, using the PathSetter software, it's a very, very simple analysis. Now, when you use this, this particular solution, what we have had shown, when we first launched the product, is that you get very, very good reproducibility for your different cell populations with this product. And what we showed initially was if you took the same PBMCs or whole blood and you ran it at 3 different sites, so on 3 different instruments, as well as having 3 different operators at those different sites, so basically 9 different conditions, you got very, very tight cell populations. So CVs typically of less than 1%. The only time when you get CVs really greater than 1% is when you have very low population percentages. And in that case, the overall change is less than kind of 1% for that cell population. So it's very reliable and compared to traditional flow cytometry. And I've shown that for the 37 different cell populations here. What's also important to note here is we have been asked by a lot of folks is, okay, so if we're doing a very, very large study that's going to take several years and we have different lots of antibodies, what will the variability be? So we actually took 10 different production lots and took the same whole blood sample, ran the 10 different lots, and you can see again, you get very nice variability. So this is a very, very robust product across sites, across operators, and across lots that is important for a lot of the large scale clinical studies that are being undertaken, especially in the area of what's going on with, with the pandemic. The other thing that's interesting to note that I'm not showing the data for here is that if you stain your cells, with the MaxPAR direct immune profiling assay, and then essentially freeze the pellet, and then run all of the samples later at a site. So for example, if you were doing a study where you were collecting samples, say, across the DC area, and you knew that you were going to do all of the analysis at Georgetown, you can actually freeze the stained pellets. And again, you will get the same type of very, very tight cell population distributions across that protocol. So this is a very, very robust platform. We just recently, because of some of the needs and requests that we've received, have developed some additional panels that can be used with the MaxPAR direct immune profiling assay. Unfortunately, at this point in time, the drop-in expansion panels are actually liquid. So it's a little bit harder to use in that it's multiple steps when you do the staining, but you see the same types of results. This is the advantage of this technology, is that with the original 30 markers, one can very easily add 8 to 15 additional channels, depending on the biological questions that you want to answer with your study. What I'm showing here is the results of a paper that came out of China very, very shortly after after SARS-CoV-2 started moving through the population. This is a relatively old paper that came out in the early part of 2020. And it just so happened that this group was looking at different strategies to improve outcome of patients. And they had a system in place where they could essentially knock out the ACE2 receptor in the mesenchymal stem cells of individuals. And what they were able to demonstrate was that you can really improve the outcome of patients with severe SARS-CoV-19 pneumonia. This was published very quickly. They were able to use a 36 marker CyTOF panel, and they were able to really demonstrate the mechanism by which these mesenchymal stem cells were able to improve the outcome and reduce the systemic inflammation in the individuals that were infected with SARS-CoV-2. We've had other customers looking at this in the general context of infectious disease. We're really I chose these examples today based on what I thought was the interest of this particular group. And this was the use of CyTOF for looking at vaccine efficacy. If you go to our website, you can actually see there are, we have a large bibliography of different types of studies that are used in different parts of infectious disease using CyTOF. And so this was a paper that's about 2 years old, where they really looked at the diversity of the CD4 and the CD8 T cell populations before and after vaccination, both in children and adults, to try to understand why the vaccine wasn't working as well in children as adults. And so what you see essentially are the typical Tisney plots on the right, where you can enumerate the different cell populations from this relatively large plex cytometry experiment. What they were able to do, and you can see the data here, and is they were able to identify actually very large differences between the CD4 and the CD8 T cell populations in these different groups of individuals, based on age. And so this is an explanation, of course, potentially for why there is the reduced vaccine efficacy. So it's really driven by the cellular response. Now, of course, how they actually take this information and then modify the vaccine is the next step in the process and what they're working on. And there's other papers that our clients or our customers have used, where they do things like look at the role of the adjuvant in some of in measuring the immune response for the particular vaccine that they're working on. And so there's a very rich amount of biology that one can extract using CyTOF technology. And so that's one of the large advantages that we see, is because you can ask very general questions about the overall immune system, not just focusing, say, on a small subset of the immune cells that may be playing a role in the vaccine efficacy. So now I would like to turn the discussion to the use of imaging within mass cytometry. And so this is the same slide I showed earlier. And because we're using labeled antibodies, you can imagine that the technology is very similar to what you would do in a standard immunofluorescence experiment, where instead of, again, using a fluorescently labeled antibody, we're using metal labeled antibodies. And especially in tissues like that are formalin fixed, autofluorescence can often be a problem. And so you actually see additional advantages with the technology here than you even do in flow. The way we actually turn this into an imaging platform is we have the imaging module that comprises the Hyperion system when you combine it with the Helios, which is that kind of rectangular box in front. And essentially what we're doing is we have a laser ablation unit within that box. And what we have in the box is the laser and the slide. And you essentially will have a laser that has an approximately 1 micron diameter to it, or a 1 micron diameter spot size to it. And then you'll come in and you'll basically laser ablate that material off of the slide. And then that will actually be transferred through a plastic tube into the Helios mass cytometer. And so the information that you get from that one piece of tissue that moves in is you are able to measure all of the ions that are on that tissue, which of course are correlated with the antibodies. By rastering the laser across the tissue, you can then create an image with a resolution of 1 micron. And what we're showing on the right are some very simple 4-color or 5-color measurements off of a slide that allows you to actually create an image from this data. And so again, this is very similar to what you would see in like a digital camera where it has a certain pixel size. The pixel size here is determined by essentially the laser size. And then you reconstruct that just like you would in a CCD camera where you're counting metal ions rather than counting photons. So one of the things that we have done, we haven't released quite as many antibodies and full solutions for the imaging platform as we have for the suspension-based platform. And there are a few reasons for that. One of course is staining of fresh PBMCs and fresh whole blood is a lot more uniform than it is in FFPE tissues, especially with the different conditions at which the slides were prepared or those blocks were prepared. But one of the things that we recently released was our cell segmentation kit, which I think significantly improves the quality of the data that one can get out from these types of analysis. And so what we have done is we've created a 3-marker panel that allows one to better elucidate the cell membranes, which is important for the single cell analysis, which I will now show you. This just is some of the highlights of the cell segmentation kit. We're combined with our iridium-based dyes for determining the nucleus. The demarcation of the plasma membranes really facilitates the single cell analysis. And for this group, this may not matter since this is a relatively new technology, but we were able to use 3 new metals for this. So if you have an existing panel or an existing workflow, you can easily add this to this without disrupting your existing panel. This just shows the 3 channels. These are 3 different isotopes of platinum that we use. And what I'm essentially showing here is, with these 3 different markers, you get very nice staining across this particular tissue. And for the folks that were kind of experts in the technology, one of the questions they had is, how does this new chemistry that we're using for the platinum conjugation compared to the standard chemistry used when we use the lanthanide metals? And you see here that you get essentially equivalent results. And so it's nice that we were able to add additional channels to the catalog. So here is an example, and unfortunately I can't tell you what tissue this is, but if you look at the results with the cell membrane staining along with the DNA channels, And using the what we're calling an optimized pipeline for doing the cell segmentation and the identification of the single cells, you see that the latest algorithms that are actually they're described in this document in the end with the references really gives significantly improved results. And if you look at this carefully, you'll see that the the green lines essentially are the outlines and the boundaries of each of these cells. And if you look at the view on the right, it actually has a structure that looks much more similar to what you're used to looking at when you're looking at an epithelial tissue, right, within a under the microscope, compared to what you're seeing on the left. For example, you can see the ductal structures that are just not correctly represented when you use a very traditional, simple end distance clustering method. I'm sorry, cell segmentation method. And the reason this is really important, and I'll show you why in a minute, is typically the way we analyze these types of data sets is we will go into the image and we will try to identify single cells. And then in each of those single cells, we'll do something very similar you would do in a flow cytometry experiment and determine the relative protein expression in each of those cells, essentially by summing up the intensity of all of the pixels that are within that cell. So it's important to identify, it's important and critical to figure out how you assign, what each pixel to what cell. And so obviously when you have a picture like either the one on the left or the right where you have a single cell, you can basically just count up every pixel within that boundary, and those boundaries obviously will give you a very, very different picture of what the single cells are, based on how you've drawn those boundaries. And so, for example, kind of when you look in the center of the picture on the left, the cells are much larger, and if you look carefully, I'm not sure you can see it on your computer, but unfortunately, there are often several nuclei that are within that single cell when you use a very, very simple end distance method. The optimized pipeline uses a combination of tools that are publicly available. It uses a tool called Elastic, which is essentially a random forest classifier to help enumerate what each pixel is when you're doing the cell segmentation, whether it's a membrane, whether it's nuclei, or whether it's cytoplasm. And then you use Cell Profiler to actually propagate out from the nuclei to the cell boundaries. And that's one reason why the cell segmentation kit significantly improves things. So this is an example of another tissue where we use the cell segmentation kit, but we use two different versions of the algorithms for doing cell segmentation. And what I really want to show you is that, depending on how you, do the cell segmentation, you can get very, very different results in your data. So, the first thing I think that you'll notice on the differences in these images is you don't see that the cells are kind of contiguous through.
I think we lost the speaker.
Yeah, looks like, yeah.
Andrew is back. We cannot hear you. You have to unmute.
Got unmuted. I don't know what happened. Everything just flashed and it said I'm being recorded. So I'll come back. I apologize for that. I'm not sure what's happening. Maybe it's a bandwidth issue today. I'm not sure. Okay. And can you see this now?
Yeah.
Okay.
Not yet.
Not yet?
Not yet. No. Yeah.
Okay.
You're fine.
Okay, good. Oops, I'm sorry, this is out of order. So, This is the, okay, so after you do the cell segmentation, you can create something that looks like an FCS file, from the data, because you've integrated all of the intensities. And then you can do, your favorite type of single cell analysis. And so what we've done here is we've done a simple phenograph. And, we identify the different cell types, we assign a color to each cell type, and then we go in and color each of those cells. And so with, with two different images, okay, which are actually from the same tissue type, what you see is, if you just look at, let's just focus on the left panel for a second. You see those open areas, and you see that the region on the left, you have a lot of these orange cells, okay? Now, when I look at the next tissue, okay, which actually has the same morphology, it's the same disease type, if you just were to look at it using, an H&E stain, the cells that are green in the upper right-hand corner are actually very similar to the cells that I had labeled as orange. And when I've done this phenograph typing, my conclusion would be that they are very different cells. And if you look at the different colors within this, you don't see a lot of commonality of the different cell types within that tissue. So your first conclusion would be that, oh, these are actually two very different molecular subtypes of that, of that tissue, because either it's, a different disease, or maybe you've looked at a region within that tissue where you don't have either the infection, or you've chosen like a non-cancerous region. Unfortunately, I don't recall exactly what these specimens were. Now, when you look at it using the improved cell segmentation, or what I'm arguing is the improved cell segmentation, if you look at those cells that are labeled as orange here, you see that they're in this depiction, they're the dark blue and the dark red cells with some infiltration of those lighter green cells. And then if I look at the other tissue now, which was in the upper right in the green, you see it has that same morphology. And you see that the overall cell types that exist within these two different tissues are actually very similar. And so the results that you have when you interpret this data can vary very widely depending on how you're going to actually analyze the data. And so we have spent a lot of time trying to figure out the best way to do this, looking at these, these are just kind of two examples of this over a wide range of tissues. And we believe that we found a way that gives, at this point at least, the most robust results. And so when you do a kind of a typical clustering, with this type of data, what you will typically get out is the phenograph heat map, which will show you for each of the different cluster types, kind of the relative expression of those different markers. And then when you look at a nice tissue like this, where you can really recognize the types of features you'd expect to see within the tissue, you get things that actually make a lot more sense in terms of, okay, these are all the same cell types. I see the same duct-like structures throughout these different tissues when you do this type of unsupervised clustering analysis. So I'll just quickly show you a paper that came out earlier this year. This was looking at kind of the spatial landscape of lung pathology during COVID-19 progression. This was, you can kind of see the thing. They had, unfortunately, very few samples. Unfortunately, I think, Marta and I spoke about this actually at one point in time in doing a similar study. But access to these tissues is very difficult because most of these lung tissues that were accessible were actually from autopsy. And with SARS-CoV-2, there were a lot of difficulties in getting this. And you really see the differences in the individuals that had early and late death in terms of the amount of infiltration, overall inflammation in the lungs that were associated with, with SARS-CoV-2. But of course, unfortunately, you can see this was only 6 patient samples where they looked at a large number of regions of interest that were across the cell, and these were typical from from this study. We've also worked with another customer, and this is actually in progress, where he's actually looked at differences, not just between early and late death, but also looking at infection from other upper respiratory pathogens, and looking at the differences in the amount and the type of the immune cells that infiltrate the lung under these different with these different pathogens. And this is something that we hope to publish soon. With that, I think I will stop and answer questions and allow time for Ivana to share the nice results that she has done in collaboration with Fluidigm, where they've been able to utilize the RNAscope technology, as well as looking just at antibodies, using this technology. So thank you, and I'll take any questions.
Thank you, Andrew. Does anybody has a question for Andrew?
So for the cells alone and the tissues, the antibodies so you have already optimized the kits, so right? So is there a kind of a way to custom made specific kits, or to use antibodies that maybe recognize a protein which is not in the panel but you are interested of, and it can be added there?
Right, so we have two ways we can help you with that. One way is we sell metal labeling kits, and so if you have an antibody in your lab that that works, we can sell you a kit to allow you to label. And it's very simple. Well, I mean, depending on, from my point of view, it was very simple, because before I came to Fluidigm, I did a lot of mass spec, and there we did a lot of things with essentially the molecular weight cutoff filters. Because that's the way we basically will do the labeling. You'll do it in the filter, and then you do your multiple washes. And then, of course, you will then have your labeled antibody in the end. And so it just takes a little bit of care, and it takes some time. The other things that we have is we have a couple of service labs that will conjugate antibodies for you.
I wonder, what machine do you need to do this analysis? Is it just regular mass spec, or?
No, this is using so we have two versions of the instrument currently on market. So we have the Helios and the imaging module, which is the Hyperion. So you can't use a regular mass spec for this. You have to use the Fluidigm instrument. And that gives you the capability of doing both imaging and suspension. We just recently released a new instrument that currently only supports suspension, and that's our CyTOF XT. It allows a little bit greater throughput on the flow side, and it's a little bit easier to run. It has a carousel for auto sampling and so forth, but you can't run this on a typical mass spec. So this uses a combination of ICP-MS coupled to a TOF, and the founders of the technology had spent years optimizing the sensitivity of the mass spec. And this is probably the most sensitive ICP-MS that's available. And so that's why you can't just use the standard mass spec. Plus all the fluidic controls up front, of course, for doing single cell analysis. You would never put really that type of wet sample, into a lot of instruments, because of all the things like salts that are in those samples.
Anybody else has a question?
Yeah, so I mean, this is for core, right? So my question is, do you already have antibodies against HIV proteins, or any other viral pathogens? I mean, apart from the host. I mean, looking at the host.
So Fluidigm does not have any. So typically, what folks will often do, if you want to look at in imaging, the easiest way to do that is to do a dual staining and use your standard antibody against that pathogen, and then use if it's fluorescently labeled, we can come in, or if it's a mouse antibody or a goat antibody, right, we can use a metal labeled anti-goat, or we can actually if it's a fluorescently labeled antibody, say, I think it's FITC, I think we have an anti-FITC antibody. And so then you can basically do a two-step staining and then stain and identify your pathogen that way. And then also then do your other staining on top of it, because of course, you'd have to use the secondary first and then come in with all of your other labeled primaries afterwards.
Okay, thank you.
You're welcome.
So, and Andrew, do you do the metal conjugation of the antibodies for us? How expensive it would be?
I don't remember the price offhand. We're actually launching a new service. I think it's typically, I want to say a couple of $1,000, you know, for that service. But I don't remember. I also know that it decreases significantly with the number of antibodies that you need labeled. But we're constantly adding antibodies to our catalog as well.
Okay. Okay.
So I have a quick question regarding the cell suspension, Andrew. You mentioned that you can collect the blood from the patients and put the cocktail of antibodies and freeze the cells. So how you do the exclusion of dead cells after that?
How do you do what after you freeze?
Exclusion of dead cells.
Oh, so we have a live dead cell staining in one of our channels, and so we can basically exclude dead cells during the analysis. They actually get run through the analyzer, but we have a live dead stain.
So you have to put everything before you freeze the cells, or how you do that?
Well, no, you just freeze the cells, and then during the analysis, we like the data I showed you, that was all based on just the live cell counts. And so you will have dead cells that make it into the cytometer, but you exclude those because you have a live dead channel as well.
Yeah, thank you.
You're welcome.
So, and just another quick question, because you know, it's an HIV field that they are trying to look at transcriptionally active viruses. What will be the minimum, do you think, how many cells you can reliable get expression of these viral proteins in PBMCs from people with HIV? What do you think will be the sensitivity to detect those very small transcriptionally active cells?
That's a really good question. In terms of the sensitivity, I'm not sure I know the answer to that. I think I had to sit down and talk to you a little bit longer to try to better understand how we would do that. I wouldn't want to guess, because it would just be a guess at this point without talking to you a little bit longer about the mRNA.
Yeah. Okay.
As a quick follow-up, for example, you are running a sample, how many cells normally you need to run?
So we typically collect on the order, well, so again, it depends, right? So if you're looking at something where you're looking at a very rare cell population, right, you're going to need to collect a lot more events, right? And so typically with the example I showed you with the MaxPAR direct immune profiling assay, you know, we typically collect, you know, around 500,000 events.
Okay, that's reasonable. Thank you so much.
You're welcome.
So if anybody else doesn't have any question, we are gonna move with Ivana's. Ivana Peran is an assistant professor at the Lombardi Cancer Center at Georgetown University, and she's using this technology, and she's gonna share also what would be the pipeline that if anybody is interested to use the technology, she will be your person of contact.
Thank you, Marta, for the introduction.
Thank you, Ivana, for being here.
Of course. So I'm going to start sharing my screen. Do you see it fine?
Yes.
Okay, good. So as Marta mentioned, I'm here at the Georgetown and I manage imaging mass cytometry. And today I want to present briefly workflow and application of IMC technology. So first, very briefly, I want to introduce our shared resources here at Lombardi. And here you see the list of many shared resources that we have at Lombardi. And there is also a link to the website where you can learn more about each one of them. However, today I'm going to focus specifically on mass spectrometry and analytical pharmacology shared resource, and also on histopathology and tissue shared resource. All of these resources are directed by Dr. Steve Byers, and our administrative director is Dave Gorlitz. So Hyperion system that I'm managing is a part of proteomics core, which belongs under mass spectrometry and analytical pharmacology shared resource, which is managed by Dr. Jiangfeng Ma. And here you can see his contact information as well. So now I'm gonna focus on the IMC workflow that we established here at Georgetown, and I'm gonna go through all of these steps with an example from my own research. So first, maybe the most important is to set a scientific question or hypothesis that you want to ask. And here I'm gonna give you example from my own research. So I'm studying pancreatic cancer for the last 10 years. And to do that, I'm using a genetically engineered mouse model of pancreatic ductal adenocarcinoma, or PDAC. So I'm using KPC mice that have mutation in KRAS and P53 genes that are also very common in the human disease. We are specifically interested in stroma of pancreatic cancer, which is very abundant, and it can comprise up to 90% of tumor mass. This stroma is consisted from activated fibroblasts. And through our research, we realized that cadherin 11, which is an adhesion molecule that is expressed on activated fibroblasts, is also increased on PDAC samples from human and mouse tissues. Specifically, cadherin 11 is expressed on cancer-associated fibroblasts, and it's not expressed in normal pancreas. So here I use this KPC mouse model that has different expression of cadherin 11. So in black line, you see KPC mice that express 2 wild-type copies of cadherin 11. And you see their survival is about 5 months. Median survival is about 5 months. And mice, KPC mice that are heterozygous for cadherin 11, they survive significantly longer. This was very interesting observation, and we decided to move forward with gene expression analysis of those tumor samples. And here we found unexpected result that was increase in antigen processing and presentation genes when mice are deficient for cadherin 11. We also saw increase in markers that are associated with dendritic cells, M1 macrophages, and B cells. And as well, we saw decrease in markers that are associated with immunosuppressive cells, such as M2 macrophages and the myeloid-derived suppressor cells. So obviously, this led us to our scientific question, which was if survival of our KPC PDAC-bearing mice with different cadherin 11 status correlates with changes in immune cell infiltrates within tumor microenvironment. So to answer this question, we wanted to do imaging of tumor samples. First, next step was to obtain tissues, in our case from mice. And here you see brief workflow. I isolated cancer from the mice, I formalin fixed them, and then our histopathology and tissue shared resource did paraffin embedding and sectioning. Next was to select antibody panel for multiplex staining in respect to our scientific questions, and also in respect to conjugated metals on each of those antibodies. Here I'm showing you the table with a list of antibodies that we used in our recently published gastroenterology paper. And here you can see that we used CD4 markers, CD8 and CD19. As well, we use marker for tissue structures. So we used E-cadherin for cancer cells, and we used alpha-smooth muscle actin as a marker of activated fibroblasts. We also used DNA intercalator. Next step, which is very important, is to confirm that your antibodies of interest are specific for the protein that you want to study. And Fluidigm recommends to test this by immunofluorescence on tissue of interest. So I did some immunofluorescence studies, first on human PDAC cancer-associated fibroblasts. And I showed that antibodies that we are using are specific for the cells that are expressing these proteins. But also another option is to use the data that you perhaps have from fluorescence imaging, such as Vectra. And our HTSR resource also has Vectra systems. So we already knew that these markers, for example, CD8, FOXP3, CD19, CD68, etc., are working fine, properly as they should. So we use that information when we are building our IMC panel. Next step is to obtain mental conjugated antibodies, and there are two routes to do that. One route is to order them directly through Fluidigm, and those antibodies would already be conjugated with the metals. Or the other route is to order other antibodies from other vendors that would be carrier-free and glycerol-free. And then you should just follow the simple protocol shown on the right side to conjugate antibodies to the metals. So each lab can do that individually, or HTSR is going to offer this also as a service. Next step, obviously, is finally multiplex staining of the tissue sections of your interest. And this will be done through HTSR as well, because the staining procedure is very similar to IHC procedure. Our HTSR contact person would be Aaron Rosenboom, and you can see here his contact information. Next step, after the staining is finished, which is very important, would be selection of regions of interest. So maximum ROI size that's recommended by Fluidigm is 1.5 millimeter square. However, if you need to image larger areas, we could set up ROIs that are next to each other. So you can select your ROIs after the multiplex staining in a way that you would use consecutive H&E stained section, and then label ROIs of your interest on a whole tissue scan. And you can see an example here below. Alternatively, you can select your ROIs before multiplex staining in a way that you would create tissue microarray. So on the left side, I already labeled ROIs. And in that case, I can overlay my H&E section with the tissue block. And then we can create a tissue microarray. What is interesting with this, that actually the core size in tissue microarray is approximately the ideal size for imaging. Finally, after your tissues are stained, I'll perform imaging on Hyperion, as Andrew already mentioned. Resolution is 1 micron. Scanning speed is about 1.5 hours per millimeter square, and the maximum continuous imaging time is about 72 hours. So this is the figure that was produced in very good collaboration with Fluidigm, specifically Andrew. and Anna, who did the imaging of my pre-selected ROIs at Fluidigm. Finally, the last step would be data analysis. And the first step in data analysis is opening your images, your MCD files with MCD Viewer, which is available from Fluidigm for free, and it works on any PC. After you open your MCD files in MCD Viewer, you can export your images as TIFF or JPEG files. And then you can use those for your downstream analysis. And there are plenty of free options such as ImageJ, Cell Profiler, Histocat, or QPath, and also some commercially available options such as Halo and Visiopharm. So on the right side, I used very simple analysis method. And here I just quantified numbers of CD4, CD8, and CD19 cells in the tissues obtained from KPC cadherin-11 wild types versus heterozygous mice. And I saw increase in immune cells in KPC cadherin-11 heterozygous animals. As a conclusion, with a huge help of Fluidigm and imaging, we showed that loss or inhibition of cadherin-11 changes this microenvironment of pancreatic cancer from immunosuppressive towards anti-tumor immune environment. And this eventually translates into increased survival of these animals. And lastly, I just want to acknowledge our Lombardi Cancer Center support grant that was awarded to Dr. Lou Wiener. Obviously, I want to thank HTSR, specifically Aaron, Winona, and Anju, our animal model shared resource. And I want to say a big thank you to Fluidigm for allowing me to use their Hyperion system before we headed to Georgetown. And here are the other funding sources as well. So now if you have any question, I would be happy to answer.
Thank you, Ivana. Any question for Ivana? So, Ivana, I remember talking with Andrew at some point about combining Fluidigm system with in situ hybridization. Did you guys some experience that you can share with the HIV group that will be interested to detect proviruses or active transcriptional viruses there?
Yes, so as Andrew already mentioned, in our gastroenterology paper, we also used the IMC coupled with RNA-scope. In our case, we detected cytokine CCL21, and it worked very well. However, for now at the Georgetown, we don't offer this technology coupled together for now. Mm-hmm. So that's why I didn't put it in presentation today. But it worked fine in our collaboration with Fluidigm.
Mm-hmm. Okay, thank you. Any other question from the audience? Okay, we finished on time. Perfect. So thank you so much, Andrew, for the presentation, and Ivana to present all and share her experience with this exciting technology. I hope we can do help us to advance the HIV field, definitely.
Yep. Thank you for the opportunity to share with this group the capabilities of our mass cytometry platform, and we look forward to supporting both Ivana at Georgetown and any of the folks on this call for their analysis. Be happy to help.
Thank you.
Thank you, everybody. And if anybody has some questions afterwards, please contact me at ip62@georgetown.edu.
Thank you.
And I'll be happy to answer any questions.
Okay.
Okay. Bye-bye.
Thank you, everybody. Bye-bye.